Gregor Mendel is considered the father of genetics, so it's fitting that our first video of our genetics unit features Gregor Mendel, and this is video 11.1 about Mendelian genetics. This video, along with the problems we do in class, will help you accomplish these goals. So if you'd like, pause the video, take a closer look. Who is this Gregor Mendel dude anyway? Well, he has almost a really cool first name, but he was an Austrian monk who lived in the 1800s, and he made his contributions to genetics using pea plants as a model organism. He was able to make his conclusions because he just had massive amounts of data. The data in his experiments were meticulously documented and organized. So, in short, though, Mendel is considered the first to describe the mechanism of inheritance, or how traits get passed from parents to offspring. Gregor Mendel manipulated the reproductive anatomy of pea plants so that he knew exactly which plants were fertilizing which. To understand what Mendel did, we have to know a bit about plant reproductive anatomy. In our photosynthesis unit, we learned some plant parts that had to do with photosynthesis, but all of these new parts discussed here are specific for plant reproduction. The main reproductive part of a plant is a flower. A flower has both male and female reproductive parts. Like in other sexually reproducing organisms, sexual reproduction in plants has to do with sperm fertilizing an egg. In plants, sperm are contained in grains of pollen. So strangely, when there's pollen in the air, you are breathing in plant sperm. And that makes you think different about your pollen allergy, doesn't it? Anyway, pollen is made and held by another plant part called the stamen, which is this whole part right here. You can tell the stamen is a male part because it has men in the name, and that's a good mnemonic. A specific region of the stamen that holds the pollen is called the anther, and you can see that blown up right there. Usually the anther is positioned so that the pollen will stick to any pollinators that come into the flower that have to go way down here to drink the nectar. You can tell that the anther is a male part because men think they know all the answers to all of life's questions. Ha! Dad joke. Now onto the female parts. On the same flower as the male parts is the ovary of the flower, shown here as a zoom in from what's going on here. The ovary contains the ovules, or eggs. Once fertilized by sperm, fertilized ovules will become the seeds, and the ovary will become the fruit. A part of the flower that helps catch the pollen is called the stigma, up here. The stigma often has a sticky fluid on it to help increase the chance of catching some pollen from the air. Once pollen binds to the stigma, the pollen travels down this part called the style to get to the ovules to fertilize the ovules. Of course, you know why it's easy to remember that the style is a female part, because the female's got all the style. Because plants have both male and female parts, plants can self-pollinate. Technically, self-pollination is not asexual reproduction because self-pollination involves sperm and egg cells, but self-pollination will produce plant offspring that are nearly identical to the parents. Plants can also reproduce by cross-pollination between flowers of different plants. Knowing about plant reproduction, Mendel could control which pea plants mated with which by removing the anther of plants so those plants couldn't self-pollinate. He took away all the pollen. In the experiments that Mendel is most famous for, Mendel first made pure breeding parent plants by allowing the plants to self-pollinate for several generations. These parent plants are called the pea generation, where P stands for parental. Then, Mendel would cross-pollinate two pure breeding parent plants to create offspring. The offspring are called the F1 generation. Mendel would then let the F1 generation self-pollinate to create plant grandbabies called the F2 generation. One of Mendel's most famous experiments examined flower color in pea plants. He cross-pollinated pure purple and pure white parental plants. This resulted in F1 plants that were all purple. After letting the F1 plants self-pollinate, he found that the F2 generation had both purple and white plants. This was really surprising, and I bet Mendel probably thought he must have made a mistake at first. But what he actually found, when looking more closely, is a pattern. Mendel examined the 7 different pea plant traits shown in this table: flower color, seed texture, pod color, pod texture, the flower positioning, and the plant height, with the same setup as the flower color. You can see that some of these experiments looked at over 8,000 plants, and what he saw was the same 3-to-1 ratio in the F2 generation with all of these experiments. The explanation that he came up with to explain his data is now known as Mendelian genetics. Even though Mendelian genetics is considered simple by today's standards, the principles of Mendelian genetics were revolutionary at the time in the late 1800s and early 1900s. Mendelian genetics deals with traits that are determined by one locus. A locus isn't an insect, that's a locust with a T at the end. But a locus is a spot on a chromosome where a gene is located. So think of locus and location of a gene. In Mendelian genetics, an individual has 2 alleles for a trait. And in Mendelian genetics, one allele is dominant and the other allele is recessive. So the rules that we've been using for genetics in our course so far. Mendel's work paved the way for us to understand the terms genotype and phenotype, where genotype is the combination of alleles in an organism, and a phenotype is what you observe about the trait that's encoded by the genotype as well as influenced by the environment. The genotypes in Mendelian genetics are homozygous dominant, heterozygous, and homozygous recessive, and the phenotypes are either dominant or recessive. Mendel's work also inspired other scientists to come up with the Punnett Square, a tool that's used to predict the genotypes and phenotypes of different breeding crosses. We could use Punnett squares to explain Mendel's pea plant flower data. To set up a Punnett square, you split one parent's alleles along the top and you split the other parent's alleles along the side. Next, you cross the alleles to simulate a fertilization, like this big pea egg was fertilized by this little pea sperm. Then what you do is you count up all of the resulting offspring. So, a pure breeding white plant breeding with a pure breeding purple plant gives these results of all heterozygous purple offspring. Now, make a Punnett square to show self-pollination of the F1 generation. Set up the parent alleles, cross the parent alleles together, then count. And this Punnett square shows a genotype ratio of 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. We call this a 1:2:1 ratio. The homozygous dominant and heterozygous offspring have the same purple phenotype, so we can combine those together. When you group those purples together, that makes 75% purple and 25% white, or a ratio of 3:1. This is how the white phenotype could skip the F1 generation and return in the F2 generation. When we talk about Mendelian genetics, we have to talk about Mendel's 2 laws of inheritance. Mendel's laws of inheritance are the law of segregation and the law of independent assortment. Let's look at each individually. First is the law of segregation. The law of segregation says that the alleles for a trait segregate into different gametes. In other words, the gametes of these plants can only have one P allele. The gametes of the purple plant will have either the big P or the little p. The gametes of this white plant will have all little p, but only one little p per gamete. Now that we're bringing some knowledge about meiosis to the table, we can connect Mendel's law of segregation back to meiosis. So, when do the alleles segregate in meiosis? The segregation happens in anaphase 1. The original cell has, say, big P and little p. At anaphase 1, well, this is metaphase 1, you see the big P and little p line up next to each other, and then in anaphase 1, the big P and the little p will segregate from one another. So at the beginning of meiosis 2, or the end of meiosis 1, the cells only have either the maternal or paternal allele. Meiosis 2 makes sure that each gamete has only one copy of the maternal or paternal allele. At the time, Mendel didn't know anything about meiosis, so we're able to make those connections, but he couldn't. Mendel's second law of inheritance is the law of independent assortment. What this law says is that the segregation of one trait doesn't affect the segregation of any other trait. In other words, just because a gamete gets one paternal allele doesn't mean that the gamete will get paternal alleles for all of the genes. There will be a mixture of maternal and paternal alleles. The critical type of study Mendel used to discover the law of independent assortment was one called a dihybrid cross. A dihybrid cross is a genetic study that looks at two traits. These two traits in Mendel's critical experiment were seed color, yellow or green, and seed texture, round or wrinkled. Just to make sure we have some terminology straight, a monohybrid cross is a cross that just follows one trait. And just like when we looked at flower color, a dihybrid cross follows two traits. You can see that the Punnett Square gets more complicated down here than over here, but we'll talk about that more in class. So Mendel developed pure breeding yellow-seeded, round-seeded pea plants and purebred green-seeded, wrinkled-seeded plants, and he bred those two together with cross-pollination. All of the resulting F1 offspring had yellow and round seeds. So what happens when you let those F1 plants self-pollinate? Will yellow always stay with round and green always stay with wrinkled? Or could you have yellow wrinkled seeds and green round seeds? The simple answer is that the F2 generation had all of these possibilities in Mendel's data, but it's a little bit more tricky to explain why. Let's look at the two possibilities. First, what would happen if the loci or the genes assorted together? Well, this would mean that a gamete that gets the yellow seed color will always get the round seed color assorting with it. Likewise, getting the green seed color will also mean that the wrinkled seed texture would assort with it as well. With independent assortment, however, having the yellow seed allele in the gamete doesn't influence if a pollen grain will have the round or wrinkled allele, yellow round and yellow wrinkled. All four combinations are possible: yellow round, yellow wrinkled, green round, and green wrinkled. Let's check out how each of these would look on a Punnett square. If you count the possibilities on the left with independent assortment, you see a ratio of 9 to 3 to 3 to 1, with 9 yellow round, 1, 2, 3, 4, 5, 6, 7, 8, 9, 3 yellow wrinkled, 1, 2, 3, 3 green round, 1, 2, 3, and 1 green wrinkled, 9 to 3 to 3 to 1. If you count up the possibilities on the right, you see a ratio of 3 yellow round to 1 green wrinkled, 3 to 1. If we compare those expected results of those two models to what Mendel actually got, which is what's seen here, you can see that Mendel's data was pretty close to the 9 to 3 to 3 to 1 prediction of the Punnett Square on the left, with independent assortment. So this confirms that seed color and seed texture assort independently and don't influence one another. Let's make some connections. Let's connect the law of independent assortment to what happens in meiosis. Remember, the law of independent assortment says that different loci separate into gametes independent from one another. Another way to think about this is that non-homologous chromosomes align independently. Now, hopefully this sounds familiar. When does this happen in meiosis? During metaphase 1. Just because this red chromosome lined up on the left doesn't mean that all of the reds have to line up on the left. This one did, but it definitely didn't have to. So let's recap. This video started by talking about specialized reproductive anatomy of flowering plants. Flowers have male and female reproductive parts. Gregor Mendel understood how plants reproduced, and he used this to do controlled pea plant breedings for studying genetic inheritance patterns that are now called Mendelian genetics. The work of Gregor Mendel led other scientists to develop Punnett squares. One of Mendel's laws of inheritance was the law of segregation, which says that there's one allele per gamete. So when a sperm fertilizes an egg, the egg and sperm each contribute one allele to the offspring. That's how the offspring has two alleles. Mendel's law of independent assortment stated that traits don't get inherited together, but each trait gets inherited without the influence of other traits. I look forward to building on this simple Mendelian genetics with you throughout the rest of this unit.